So, hello students, welcome back to Vedantu CBSE NCERT solutions, where we find solutions to all the answers given in NCERT. And also, we have decided to teach you line by line of NCERT, right? Every single line of NCERT to make it a good concept clearance session for all of you, right? So, we finished the previous chapter, that is the cell, the unit of life. It has been completely over now. It was very detailed classes. We finished it in two parts, and it is over. Now we head to the next part, that is going to be the next chapter, that is cell cycle and cell division. And let me tell you students, every single year, right, every single year you will get around 4 to 5 questions directly. The same concept will be repeating every single year. I repeat again students, let me tell you this very carefully, that if you study the first two parts from me, right, I'll be doing in two parts, you will not even miss a single question. And I guarantee you that, because I have studied from the past 10 years question papers, and we know exactly where the question will be asked from. And after listening to my class, you can always go back and solve PYQs and just check your knowledge if you're able to solve all the questions or not. So with that being said, let's start today's class, and let's start it, friends. Let's start the first. Any doubts you have regarding any part, you can always ask me in the comment section, and also quickly smash that like button, all of you. Quickly smash that like button, because good reach, right? Now students, let's read the lines of NCERT and let's understand. Now, the first line of NCERT. First of all of you should know, cell cycle and cell division, every single year around 5 questions usually comes from here. 5 questions usually come from this chapter. 4 to 5 questions is general trend. Sometimes even 6 questions can come, right? If you're lucky, 6 questions will come, and hopefully you can answer them, right? So are you aware that all organisms, even the largest, start their life from a single cell? For example, take humans as an example, right? Take humans as an example. Simple example, right? All of you know, the single cell they're referring to here is the zygote. So the entire human life, right, I am right here, like, the entire large body right now, which I have right now, it all started from a single cell. It all started from a single cell, that is the zygote. This simple zygote, which is diploid in nature, kept on dividing, and it ended up in a multicellular structure. So students, our life started from a single cell, and it became multicellular in nature. Later on. Okay. Now you have wondered how a single cell then goes on to form a such large organism. That is growth and reproduction, students. Growth and reproduction are characteristics of a cell. So if they ask you, what are the characters of a cell? What are going to be a characters of a cell? You're supposed to me, the first one is, a cell will always show growth, a cell will undergo reproduction. That is basically cell division. Okay, so the two characters of a cell is nothing but growth as well as reproduction. The two main events. Okay, now all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells. That is, one cell will give rise to two daughter cells. Okay. Now these are newly formed daughter cells can themselves grow and divide. So these two cells can also grow and divide into two cells. Eventually, every single cell can grow and divide. Okay. So all of you remember, right? The cell theory. In the cell theory, we learned that Radolf Virchow mentioned that every single cell comes into existence from a pre-existing cell. I repeat again, every single cell comes into existence from a pre-existing cell. So this cell came into existence because of this cell, right? Now, gives new cell population formed by growth and cell division. Division of a single parent cell and its progenies. In other words, such cycle of growth and division allows a single cell to form a structure constituting of millions of cells. That is, the reason we have this multicellular organism, the main reason for this multicellular organism is because of cell division. So for a single cell, that is zygote, to become multicellular structure, the zygote has to undergo cell division. Clear? Yes. Now students, now we understand something called a cell cycle. Very important, understand the cell cycle. It is very crucial because if you don't understand cell cycle, everything will be difficult a little bit. Okay, understand cell cycle. Okay. Hello. Good morning, Tanuja and all. Okay, the cell division is a very important process in all living organisms, right? So it is a very important process in all living organisms, that is cell division. Every single cell has to divide. For example, if the cell does not divide, there will not be, the organism cannot grow. Okay, and during the division of a cell, there are major events. Okay, there are two main events in cell division. Events in cell division. Understand the difference very well right now. The first main event is something called as DNA replication. The first event is DNA replication. Okay. The second main event is going to be cell growth. The cell will grow. The third event is going to be cell division. Cell division. Three main events in the entire cell division. Okay, the first, the DNA replicates, the cell is growing, and the cell eventually divides. Okay, it takes place in a coordinated way to ensure the correct division and formation of progeny cells containing intact genome. What is the meaning of that? Is it has to happen in an order. So when you're making a burger, do you add the tomato, onion in the outside or the inside? Obviously the inside. First you take the two buns. In the middle you add the aloo patty or chicken patty. Then you add tomato, sauce and everything. Or add the sauce on the top, then the bun. No. When you are making a burger, it has to go in an order. So even when there is cell division, it has to go in a proper order. Okay. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into total cells is termed as cell cycle. So what is cell cycle, students? Cell cycle is nothing but the events in which the cell divides. For example, making the burger is called as the entire process. You get the buns, you cut the onions, you cut the vegetables, then you make the burger. Similarly, for a cell to divide, there are few events such as your duplicate of DNA replication, the cell growth is happening, then the cell division is happening. Right? Cell division is happening. It happens in an order. When this happens in an order, the entire order itself is called as cell cycle. It is called as cell cycle. Okay, cell cycle. That entire order is called as cell cycle. Okay. Although cell growth in terms of cytoplasmic increase, okay, very important line of NCERT which everyone misses, very important line. Although cell growth in terms of cytoplasmic increase is a continuous process. See? So if I ask you question, students, assertion reason, cytoplasmic increase is continuous process. Simple assertion they'll ask you, assertion. Cytoplasmic increase is continuous. It is going to be continuous. You'll be like, yes sir, assertion is correct. Reason they can give you something, random reason they can give you, then they'll give you only assertion is correct, reason is wrong. They can give you a question like that. But remember students, the cytoplasmic increase is a continuous process, but the cell division, when the actual replication is happening, see this, DNA synthesis occurs only during a specific stage in the life cycle. Example students, this right here, the specific phase they're talking about is none other than the S phase. Only in the S phase, students, only in the S phase, DNA replication happens. DNA replication happens. In other times, DNA is not replicating. So understand this. The cytoplasmic increase will happen throughout the life cycle. Cytoplasmic increase. But the DNA replication will happen only in the S phase. Only in the S phase. Now, what is the S phase? We will learn in the next few minutes. Understand. The replicated chromosomes, the DNA, are then distributed into daughter nuclei by complex series of events during the cell division. These events are themselves under genetic control. The entire event is under genetic control. So, what is happening, students? For example, if this is a cell right here, this is a cell right here. Within the cell, there is only one DNA first. The DNA duplicates now, can you see? There is DNA duplication. Then what happens? Then the cell will divide into two cells. And each one will have the DNA. Understood? Again, within the cell also DNA will replicate. Again, the cell division happens. The DNA will be sent to the progeny, half of DNA. Okay, that's how the DNA replication happens. This entire complex of events. So what is happening first? First DNA replication is happening. After DNA replication happens, cell division is happening after that. Cell division is happening after that. Okay. Now they have given you some few examples here. Understand the few cell division. Now something I told you about cell division. That is the time required for the cell to divide. Now this time can vary. This time can change for different organisms. For example, for a bacteria to divide into two daughter cells, it just requires 20 minutes. That's all. In just 20 minutes, a cell will divide. For example, for yeast, it requires 90 minutes. 90 minutes. Now, what about for a RBC cell? For RBC cell to divide, it requires 24 hours. 24 hours for a red blood cell to divide. A human cell to divide. So, for a cell to divide into two daughter cells, it requires a lot of time. Okay. A typical eukaryotic cell cycle is illustrated by the human cell in culture. Now, these cells divide once in approximately 24 hours. See, the RBC cells, human cell, basically. Okay. However, this duration of cell cycle can vary from the organism to organism and also from the cell type to cell type. Yeast, for example, can divide in 90 minutes. Bacteria in 20 minutes. Okay. Now, students, you need to understand the entire cell cycle. The entire cell cycle is broadly divided into two categories. The entire cell cycle is divided into two categories. The first one is called as the interphase. The first one. Second one is called as the M phase. First one is interphase. Second one is called as the M phase. Let me explain a very simple example. The difference between interphase and the M phase. Understand? Right now you are preparing for NEET examination or board examination. Okay, preparing for examination. For the examination preparation, you will spend one entire year. For preparing for the examination. Right. Now, once the exam comes, that is going to be the main exam is going to be on someday for 3 hours 20 minutes if you are writing NEET examination. For writing that 3 hour 20 minutes examination, you are preparing for 1 or 2 years. Yes. Your interphase is like the preparation phase. It is like the preparation phase. All of you right now are in the interphase. Preparing for the examination. In the case of cell cycle, the cell is preparing for the cell division. The preparation stage for a cell also is called as interphase. So in the interphase, students, the cell is preparing to divide. It is not dividing. The cell is preparing to divide. Like how you are preparing right now for the examination, the cell also is preparing for the cell division, and the preparation stage is called as interphase. So if the cell is preparing in the interphase, where the cell is actually dividing, then the cell is actually dividing in the M phase, also called as mitotic phase. This mitotic phase is like your exam. You prepare right now, then you give the examination. So see, this M phase is the mitosis phase. In this exam phase, where what happens? The actual cell division happens here. Cell division. Interphase preparation, M phase cell division. Is the difference clear to all of you? Tell me. Is the difference clear to all of you now? Simple difference. All of you right now in the interphase. The day you give the exam, you will go to the M phase. Similarly, a cell is preparing for the cell division. A cell will reach M phase. Only when it reaches M phase, it will undergo cell division. Done. Nishad, if I speak Hindi, your ears will bleed. My Hindi is so bad. I will mix up genders. I will tell half the words I don't know. Right? I'm still learning, so it is something new for me. I'm trying to learn Hindi, but it is a little difficult because all your life you don't speak Hindi, and suddenly when you try to learn Hindi, it becomes a little difficult, right? So I'm trying to learn Hindi, but it is a little difficult. Okay, someday, right? Someday I can teach Hindi in Hindi. So let's hopefully that day comes soon, right? Trust me, and asked my other students if they have heard me speaking Hindi, they'll go, even they're like, sir, never speak Hindi in your life. Even they have already told me. Okay, so someday we'll see. Someday, not now, not now. Forget mitosis. You will not understand what I'm speaking only. Okay. So forget cell cycle. You will not understand what मैं क्या करता हूँ बोलता हूँ. You will get you will not understand only. So this is clear, right? Then Nishad, this is clear, right? What is interface? What is M phase? I hope that is clear to all of you. Basic. See, knowledge is important. It doesn't matter how you're acquiring it. Right? It doesn't matter how you're acquiring it, which medium you're acquiring it. All that matters is if you're learning something, right? So learn. Right, just learn. Okay, so M phase and interface is done now, right? Now students, what I told you right now is given in the diagram. For example, do you see this light blue color, students? This light blue color everywhere. This light blue color is nothing but the interface. Right here. It is the interface, light blue color. And can you see this black color part here? That is the M phase. Now do you see, the cell is around 95% time only in the interface. It is preparing nicely. 1, 2, 3 drops it is taking, okay. It is taking 2, 3 drops and it is preparing for the M phase. Can you see? First drop, second drop, third drop. Okay, it is taking long drop and it is preparing for the examination. Now here just the 5% is going to be the M phase. Okay, understand. Now students, I'll give you a quick example. Let's read what is given the next line. I hope this is clear to all of you now, right students? This is the interface, this is the M phase. The different parts of interface I'll tell you in the next slide. Or should I tell you now only? Let's understand, okay? So I told you the entire cell cycle, students. Entire cell cycle is divided into 2 categories. First one is called as the interface. Then we have the M phase. Interface is divided into 3 different categories again. Interface. First is G1, then S, then G2. 3 different phase and interface. The M phase is divided into something called as PMAT. That is Prophase, Metaphase, Anaphase and Telophase, as well as the Telophase. Okay, Telophase. 4 different phases when it comes to the M phase, 3 different phases of the interface. Now understand, only when the G2 is over, prophase will start. Only when the G2 is over, prophase will start. So we have first G1, we have G1, S, G2, then we have the prophase. Prophase, metaphase, anaphase, telophase. That is the order it is going to go. Clear all of you? This is very basic, so you need to understand. Okay. Now, the M phase. What is given here? Let's read. The M phase represents the phase when the actual cell division or mitosis occurs. I told you already. M phase is the where you're writing the examination where the actual cell division is happening. That is the M phase. Right? That is the M phase. And the interface represents the phase between the 2 successive M phase. What is the meaning of this? Meaning of this is very simple. If you go back into the diagram, can I tell after one entire interface we have an M phase? So can I tell M phase is technically between, see, we have inter, then we have M, then again we have inter, then we have M, then we have again have inter. Right. So can I tell the M phase right here, students? The M phase right here is between 2 interfaces. The M phase right here is between 2 interfaces. It's a cycle. So we have inter M, inter M, inter M. So if you go back to the slide, what they're explaining here, they're telling you here, students, it's a phase between the 2 successive M phases. Which one? The interface. So if you go see here, can I tell? Interface is between what? Interface right here is between 2 M phases. Understand? It's very simple to understand. Okay. So interface is between what? Interface is between 2 successive M phases. That's all. Now, it is significant to note that in 24 hours average duration of cell cycle of human cell. Okay, understand, this is very beautiful concept to understand. For a human cell to divide, it requires 24 hours. It requires 24 hours. Now, all of you learned that in your NEET preparation, you need at least 1 year for preparation. Board examination, little bit less time, but you have to prepare. But for a human cell, out of this 24 hours, 23 hours, it is in interphase. That is roughly around 95% of the time. A cell is in interphase. Now 23 minus 24 minus 23 is 1 hour. So that 1 hour is going to be the M phase. For a human cell, out of the 24 hours, 23 hours is the interphase, 1 hour is the M phase, which is roughly around just 5% of the time it is in M phase. Understand? So that much importance is given to the interphase. That much of time is required for a cell to divide. Okay? Cell to divide. Yes? Now, let me read what is given here, students. Pakka understood. Nice. The cell division, see, it is significant to note that in 24 hours average duration of cell cycle for a human cell division properly lasts only for about an hour. See, M phase is only for 1 hour. The interphase lasts more than 95%. Interphase is for 95%. Okay, duration cycle. The M phase starts with the nuclear division. So understand, this M phase, if I go back here, before I write prophase, metaphase, friends, can I divide this into where did it go? Karyokinases and cytokinases. Okay. Under karyokinases, can I write PMAT? Yes. Prophase, metaphase, anaphase, telophase. Under karyokinases. Okay. Under karyokinases. 1 second, students. 1 second. Can I tell that? So this total entire M phase can be divided into karyokinases, cytokinases. Karyokinases and cytokinases. Karyokinases is divided into prophase, metaphase, anaphase, telophase. Okay, beautiful. See here, the corresponding to the separation of daughter cells chromosome, karyokinases. What is karyokinases? Separation of kairo. What is kairo? Nucleus. Separation of nucleus is called as karyokinases. Okay, and usually ends with division of cytoplasm. That is cytokinases. So what is happening first? First we have karyokinases, then we have cytokinases. Okay. The interphase, though called the resting phase, is a time during which the cell prepare for division by undergoing both cell growth and DNA replication. Now you tell me, I told you have 1 year to prepare for examination. Will you sit quietly in this 1 year or will you prepare right now? Obviously prepare. Even though I told you it is called as a resting period, even though I told you you are in interphase right now, will you stay quiet and not study anything? No, obviously not. Since you are preparing for the examination, you will prepare by studying every single day, solving questions every single day, right? So similarly, students, if I call the interphase as a resting phase, it does not mean that the cell is completely resting, sleeping and eating nicely. No. The cell is preparing. It is preparing for the M phase. Okay. The cell is preparing for the M phase. So, what are the phase here? We have G1, S, G2, called as gap 1 synthesis and gap 2 phase. As we call as gap 2 phase. Any doubt, quickly ask me before we go to the next concept now. That is going to be the G1 phase. Okay. Any doubt, quickly ask me, students. Any doubt here? What is happening in G1? What is happening in G1 phase? Okay. Understand. Students, this G1 phase is also called as your post mitotic phase. Why, sir? Why are we calling it post mitotic phase? Even though this is happening before mitosis, why am I calling it post mitotic phase then? Understand? I just now told you, after G2 only M phase is happening. After G2 M phase is happening, then why am I calling G1 as post mitotic phase then? Why? Because students, it is a complete cycle. What is the meaning of that? Understand. I told you first we have G1, then we have S, then we have G2. Now what is coming after G2? Tell me, students. After G2 we have again M. Then what do we have after M phase? Again G1, S, G2, M. G1, S, G2, M again. Right? M again. So we have mitosis here. Mitosis, mitosis. So can I tell students, if you look here, after M phase what do we have? G1. After M phase what do we have? G1. Again after this what do we have? G1. So can I call it post mitotic? It is happening, G1 is happening after mitosis. Tell me, is it happening after mitosis? Yes. So can I call it post mitotic phase then? Yes. Post mitotic phase. Also called as post mitotic phase. Remember students, this M phase is also called as the longest phase. It is the longest phase. But what is actually happening here? Main work is happening here is synthesis of proteins and RNA. Proteins and RNA synthesized here. Okay. But remember students, in the G1 phase, there is no DNA replication. No DNA replication when it comes to the G1 phase. DNA replication will happen only in the S phase. Only in the S phase. See, I'll write here friends. S phase. Only in the S phase DNA replication takes place. Only in the S phase DNA replication happens. In the S phase DNA replication. And also centrioles also duplicate. Centrioles duplicate in the cytoplasm. Centrioles duplicate. Okay. So let's see what is given here. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. What is the meaning of that? G1 phase, can you see here? G1 phase, which is right here, G1. Is it between M phase? Yes, it is between M phase and initiation of DNA replication. What is the meaning of that? Initiation of DNA replication happens in the S phase. So can I tell G1 phase right now is between M and S? It is between M and S, G1. M is nothing but mitosis and S is nothing but initiation of DNA replication. So G1 phase is between M and S. Between M and S. Yes, M and S. Now during G1 phase of the cell is metabolically active and continuously grows. All of you know this now. Okay. But does not replicate this DNA. I told you already. No DNA replication in the G1. The S or synthesis phase marks the period during which DNA synthesis or replication takes place. So replication or the DNA synthesis is only happening in the S phase, not anywhere else. Okay. Now during this time, the amount of DNA per cell doubles. For example, in DNA, because of DNA replication, if the DNA content was 2C, it will become 4C. If the DNA was looking like something like this, it will become like this. 2C to 4C. 2C to 4C condition. Where? In the S phase. In the S phase condition. 2C to 4C. However, there is no increase in the chromosome number. See, this is also one chromosome. This also is only one chromosome. One chromosome. One chromosome with two chromatids now. That is the difference. One chromosome with two chromatids. So the number of chromosome will always remain here. Same. Only the number of chromatids are increasing. Number of chromatids are increasing. The number of chromosomes will remain the same. Okay. If the cell had diploid number of chromosomes, that is, if the cell had 46 chromosomes, it will have 46 chromosomes in the end. The number of chromosomes will remain the same. The number of chromatids will change. Double. Okay. Even after the S phase, the number of chromosomes remain the same, that is 2-1. In animal cells during the S phase, DNA replication begins in the nucleus and centrioles duplicate in the cytoplasm. See, centrioles will duplicate in the S phase. During the G2 phase, last phase, proteins are synthesized in preparation for mitosis while the cell growth continues. So, major of the proteins are synthesized in the G2 phase. Protein G2. Protein this is G2. Okay? Any doubt here, students? Any doubt here? Quickly ask me. I'm going to move aside so you can see the entire board. Okay? I hope you're able to see the entire board now. Any doubt in this paragraph anywhere? No doubt. Okay. Now, some cells in the adult animals do not appear to exhibit division. For example, heart cell. They tell you, no, once you give your heart to someone, it is permanent. You cannot take your heart back. Why? Because heart does not divide. You only have one heart. And just like you have one heart, the heart cell also will only be present as one. The heart cell will never divide into total cells. No. The heart cell is born, the heart cell dies alone. Okay? It cannot divide. And many other cells divide only occasionally as needed to replace cells that has been lost because of injury or cell death. So cells simply do not divide. You feel like they don't have any work. They have some work. Only when there is work, they will divide. Unless it is told so, they will stay quiet. So cells do not divide randomly like mentals. They have a specific time, their specific location, specific function, and specific destiny. Only when there is required, a cell divides. Okay? Now the cells that do not divide, for example, if there is one cell that does not want to divide, let's call this cell as Sam. The Sam does not want to divide. They're like, no. Sam doesn't want to divide. So what will Sam undergo? Sam will undergo something very special. That is, just after G1, a cell will exit. Just after G1, the cell will exit. See here. They do not further divide, exit G1 phase to enter an inactive stage called as quiescent or G0 phase. Quiescent or G0 phase. See here. So the cell is exiting after G1 and it is entering G0. Understood all of you? Understood? So after G1 phase, the cell is exiting the cell cycle. After G1, that is very important line. So after G1 phase, the cell is exiting the cell cycle and entering the G0 phase. Now you might wonder, since it is called as G0, is the cell active? Is the cell alive? Is the cell metabolically active? If it is metabolically active, can it enter again the cell cycle? All these questions I'll answer now. See here. The cells in this stage remain metabolically active. First point clear. So if someone asks you, in G0, are the cells metabolically active? You're supposed to say yes. The cells are metabolically active. Okay? But no longer proliferate. Meaning what? They may be metabolically active, but do they divide? No. That is the main important point. They are very active, but they are not dividing. They are not dividing here anymore. Okay. But can they divide later on? Yes, they can divide later on. The answer is yes. See, unless called upon to do so, depending upon the requirement of the organism. So if the organism requires more cell, the cell will come out of the cell G naught and again start dividing. Understood, all of you? Understood? The cell does not divide all the time. It is not dividing, but it is active. It is active, metabolically active. Only when there is a requirement, the cell will start to divide again. That is the beauty of G naught phase. Beauty of G naught phase. Okay. Now, in animals, mitotic cell division is only seen in a diploid cell. All of you know the mitosis only happens in diploid cell. What is the meaning of that? In the case of mitosis, we have a diploid cell. Diploid cell will divide into two cells. Both the cells are going to be haploid. So both the cells are going to be diploid, 2 to 1. There is no change in ploidy, and it always happens in diploid cell. Mitosis. Okay. But there are exceptions. For example, there are few exceptions to this where a haploid cell divides by mitosis. What is this? A haploid cell is dividing into two daughter cells. Both are going to be haploid. Haploid cells are doing mitosis. Example is what? Male drone bee. The male drone bee divides by mitosis to produce the gametes. Against this, a plants can also show mitotic cell division, that is algae. In the case of algae, a diploid cell can undergo mitosis, even a haploid cell can undergo mitosis. Both are possible. Algae are so special that both can undergo mitosis. Okay. G naught phase. It's G naught phase. The G naught phase, I'll tell you once again. G naught phase is a very special phase. Why is it a special phase? Because imagine there is one cell. Okay, there is one cell. It does not want to divide. There is one cell. It does not want to divide. So what will happen? If a cell does not want to divide, it will exit the cell cycle. For example, if someone doesn't want to study, what will they do? They'll stop the preparation. Right? I told you this G1, S, G2 is a preparation stage. If someone doesn't want to write the exam, what will they do? They'll close their books, delete all the notes, and they'll simply sit. Okay. They'll simply sit. Because they're distracted, they do not have the energy, do not have the will to study anymore. Similarly, if a cell does not want to divide, they will exit the cell cycle. Okay, they'll exit the cell cycle after G1. After G1, a cell will be out of the cell cycle. But now the cell will be metabolically active. Even though the cell is not dividing, the cell is not undergoing division, the cell is not undergoing M phase, but it is metabolically active. But imagine the cell requires a lot of energy now. The cell requires a lot of, number of cells. The G0 cell which had undergone quiescent stage, the G0 cell which has stopped dividing, it can again start dividing. If the cell has some requirement. Understood now? Tell me, understood? That is the G0 phase. G0 phase is a very special phase where a cell stops dividing, but it is metabolically active. If the organism requires the cell again, the G0 cell will again start dividing if there is requirement. So based on the requirement, G0 stops dividing, again it starts dividing. Understood? So it's given here. Some cells in the adult animals do not appear to exhibit division. That is, cells does not divide, heart cells do not divide. Right? Many other cells divide only occasionally, as when there is replace cells or have been injured. Okay, injured cells. These cells which do not divide further, a cell which is not dividing any further, it will exit the G1 phase. So after G1 phase, it will exit. It's like, no more, I can't divide anymore. But if the organism is telling, no, I need requirement, I need more soldiers, I want more cells, then the G0 cell will again start dividing. See here, the G1 phase enter an inactive stage called as quiescent stage. Inactive stage. the cells in this stage remain metabolically active. The cells may be entering G naught, but they are metabolically active still. Okay, but no longer proliferate. Meaning is that the meaning of proliferation is cell division. The cell may be metabolically inactive, but they are not dividing at all. But if the cell requires, then they'll start dividing again. Okay. So this was the line I told you. In animal cells, mitotic cell division is only seen in the diploid cell, right? In the case of animals, we have diploid cell, which will give rise to two diploid cells. This is how it happens. A diploid cell will never give a haploid cell generally. Never gives. Generally speaking, a diploid cell will give rise to diploid cell. But in the case of certain drone bee, right, male honeybee drone, a haploid cell will give rise to a haploid cell. That is, a mitosis can happen in a haploid cell. That's all they're saying. A mitosis can happen in a haploid cell, which can give rise to two haploid cells. Mitosis can happen in a haploid cell. In the case of algae, mitosis can happen in both diploid and haploid cell. Both it can happen in the case of algae. Okay. Now, with that being said, your first part of the chapter is over now. Done and dusted. Okay. First part is over now. I believe all of you understood the first part. Okay, first part all of you understood. Now we will go with something called as the M phase. Ready? Ready with the M phase, students? Tell me yes or no. Quickly tell me in the chat. Ready with the M phase? No? No. This is the most dramatic period of the cell cycle. It's like teenage. All of you teenage now. Full drama in your life. So cell is also entering a teenage now. Full dramatic phase. Okay. It involves a major reorganization of virtually all components of the cell. Like how you start fighting with everyone. Okay. There is a complete reorganization of all the parts of the cell. Okay. Since the number of chromosomes in the parent and progeny is the same, I told you there is always same number of chromosome. There is no increase in chromosome number, right? It is also called as equational division. So the mitosis is called as what? Equational. Why equational? Because there is equal number of chromosomes in the parent and the offspring. That's why it's called as equational division. Even though the cell is dividing, the number of chromosomes is going to remain the same. Number of chromosomes is going to remain the same only. Same to same. Number of chromosome. Okay. Now, though for convenience, mitosis has been divided into 4 stages of nuclear division. That is chirokinesis. There are 4 types of chirokinesis. Prophase, metaphase, anaphase, telophase. But in reality, it is very difficult to differentiate. You can't really tell which one is prophase, metaphase. It is very difficult to tell. Okay, it is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn together. So clear-cut difference cannot be told. There is always some transition phase can be seen. For example, a cell is going from prophase to metaphase. You can always the transition also you can see. So clear-cut difference is very difficult always. Okay. Now let's study chirokinesis, the prophase. All of you understand prophase now, students. Ready? I'll make you write down some points. These points are going to be helpful to you for writing the examination because questions will directly come from the points which I tell you now. Ready? Ready for prophase, students? Ready for prophase? Ready? So let's start with prophase first of all. Let's start with prophase. Prophase. Okay, prophase. Prophase, students, understand it is actually the longest phase first of all. It is the longest phase. Longest phase of all. Okay. Now what is happening here in the prophase? In the prophase there is something called as initiation. Initiation of condensation of chromosomes. Condensation of chromosomal material. Initiation of condensation of chromosomal material. The chromosomal material starts condensing. Chromosomal material. Initiation of condensation of chromosomal material. Okay. And if you want to broadly classify the prophase, it can be classified into two categories. The first one is called as the early prophase. Early prophase. The other one is called as the late prophase. Understand what is happening in early prophase first. Okay. Let's draw this. Oh, I think I erased everything by mistake, students. Let me write again now. This is the problem with the pen tab. Sometimes what happens is you entirely erase everything. Oh, I have to write again now. Longest phase and also initiation of condensation of chromosomal material. Okay. What I tell you of that? Divided into two parts. It is divided into two parts. First one is called as early prophase. And we have the late prophase. Okay. Late prophase. Okay. What happens in early prophase? This is the cell. Okay. In this cell, in the middle we have the nucleus. Within the nucleus, all the chromosomes starts to condense now. You see, all the chromosomes are starting to condense in the inside the nucleus. Apart from that, all of you remember centrosome? Centrosome, which has like cartwheel experience. Centrosome. Okay. The centrosome starts to move towards the opposite poles. This is the pericentriolar space. Okay. Now this centrosome or the centriole has two types of microtubules arising. From front, there is one microtubule called as spindle fibers. From the back, they have one more type of spindle fibers coming from the back. This is called as asters. Asters help in anchorage. Spindle fibers will attach to kinetochore and remove the centromere. Right. Spindle fibers and asters. That is, there are two types of microtubules. All of you remember microtubules, right? The cytoskeleton. There are two types of microtubules. The first one is called as asters, which you just saw. The other one is spindle fibers. Okay. This spindle fiber is present in both plant and animals. This asters is mostly seen only in the case of animals. Okay. Now, this asters plus spindle fiber is called as mitotic apparatus. That is mitotic apparatus. So what do we learn, friends? First of all, we have early prophase and late prophase. Early prophase, what is happening? There is formation of microtubules, two types, asters and spindle fiber. Aster and spindle fiber together are called as mitotic apparatus. Aster is found in the case of animals. Spindle fiber is present in the case of both plants and animals. Both plants and animals. Okay. Now what is happening in the late prophase? Okay, late prophase. In late prophase, mostly the Golgi complex, endoplasmic reticulum, the nucleolus, nuclear envelope, right? Nuclear envelope, everything will disappear. Gone in the late prophase. Everything is disappearing. That is the late prophase. Any doubt, you quickly ask me, students. It's very simple. I've made the entire notes. You cannot go wrong here. Apart from if you just read the notes, you'll understand. See, understand what is in prophase now. Which is the first stage of cheirokinesis of mitosis following the S and G2. So all of you know G1, S, G2 and M. In M, first one is what? Prophase. So after G2, what do we have? Prophase. In S and G2 phase, the new DNA molecule formed are not distinct but intertwined. Not distinct, intertwined. Prophase is marked by the initiation of condensation of chromosomal material. There is initiation of condensation of chromosomal material now. Okay. The chromosomal material becomes untangled during the process of chromatin condensation. So what is happening? The entire chromatin is getting condensed now. Getting condensed. The centrosome, which had undergone duplication, all of you remember centrosome? Centrosome had undergone duplication. I wrote here. See here, centrosome duplication. In the S phase, right? In the S phase of interphase, now begins to move towards the opposite poles. So can I write one point here? In early prophase, what is happening? First of all, movement of centrosome, right? Centrosome. The completion of prophase can thus be marked by the following events. What are the events? Chromosomal material condensation to form compact mitotic chromosomes. Chromosomes are seen to be composed of two chromatids. Can you say? I told you. It is seen something like this. Two chromatids. Why two chromatids? Because of the S phase. Attached to the centromere. The centromere which had undergone centrosome which has undergone duplication during the interphase began to move towards the opposite poles of the cell. Each centrosome radiates out microtubules. I told you. Microtubules is coming out. Called as asters. The two asters. How many asters, students? Two asters. Aster number 1, aster number 2. The two asters together with spindle fiber form the mitotic apparatus. So aster plus spindle fiber is the mitotic apparatus. Did we learn that here? Yes. Here. Together they are called as mitotic apparatus. Okay. Mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. What is it? This is characters of late prophase. Late prophase. So late prophase, everything is disappearing. Late prophase, everything is disappearing. We learned here. Late prophase. So that is the difference between early and late prophase. In early prophase, aster formation is happening. Microtubules are coming out. Asters from top, spindle fibers from bottom, Golgi complex, endoplasmic reticulum, nucleolus, nuclear envelope, everything will disappear now, eventually. And this is the end of prophase. Now, after prophase, what do we have? We have the metaphase. See, this is the early prophase, late prophase. Can you see? Condensation is done here. Condensation of chromosomes is happening. Okay. Now, what about metaphase now? Okay. What happens in metaphase? Understand the diagram, you'll understand better. What happens in metaphase now? What happens in metaphase? In the case of metaphase, all the chromosomes will align at the equator. All the chromosomes will align at the equator. Can you see? All the chromosomes are aligning at the equator. So what is happening? This is called as, obviously the chromosomes were in the sides here. Can you see? There were chromosomes were somewhere here. Chromosomes were somewhere here. The chromosomes are moving towards the this thing center now. The movement of chromosomes, students. The movement of chromosome is called as congregation. First of all, movement of chromosomes is called as congregation. Is called as congregation. Now this center part, this is called as metaphasic plate. Metaphasic plate, also called as equatorial plate. Equatorial plate. Equatorial plate. Now here what do we have, students? Apart from that, in the corners what do we have? We have the microtubules. We have the centrosome, the pericentellar space, and here we have the microtubules attaching. Let me draw it in white only. We have the spindle fibers attaching. Yes. We have the spindle fibers attaching. You see, spindle fibers are attached into the chromosome. Now you might ask me, sir, where are they attaching? What is the point of attachment? What is the point of attachment? So this is the centromere, sister chromatids. All the sister chromatids and the centromere. Do we have some protein layer here? Protein layer. The protein layer is called as kinetochore. Kinetochore. So all the spindle fibers will attach at the kinetochore. So there is also kinetochore here, students. For every single chromosome has kinetochore. So all the spindle fibers will attach at the kinetochore. Spindle fiber will attach at the kinetochore. Okay, kinetochore. Now, what is happening here, students? Spindle fibers are attaching to kinetochore. Fibers are attached to the, what happened? Kinetochore. Attached to the kinetochore. Okay, kinetochore. Now, students, tell me how many kinetochore will be there? Tell me, students, how many kinetochore will be there in the one diploid cell? How many kinetochores? How many kinetochore will be there? See, there are, in diploid cell there are 46 chromosomes. There are 46 chromosomes. Yes. Every chromosome will have 2 kinetochores. So can I tell there will be 92 kinetochores then? 92 kinetochores. 92 kinetochores. Okay. 92 kinetochore. Now the last point here is, this stage, that is metaphase right now, is the best stage to study the chromosome morphology. To study chromosomal morphology. Chromosomal morphology. Now you might ask me, sir, why? Why is the best stage now? Because, students, understand, I told you, initiation of condensation of chromosomes happened in the prophase, but the complete condensation of chromosomes will happen in the metaphase. That is, condensation is complete here. Condensation is complete here. So if the condensation is complete in the case of your metaphase, best stage to study the morphology of chromosomes. Young anther, exactly. Young anther. Right? young anther. Any doubt? You quickly ask me, students. Any doubt in the metaphase? Let's see what is given here. The complete disintegration of nuclear envelope marks the start of the second phase of the mitosis. Disintegration of nuclear envelope. By that is the end of late prophase. Okay. Hence the chromosomes are spread through the cytoplasm of the cell. Okay. By this stage, condensation of chromosome is completed. See, I told you, completed. Very important line. Okay. They can be observed clearly under the microscope. This is then the stage for study the morphology of chromosome. Best stage to study the morphology of chromosome. Okay. At this stage, metaphasic chromosomes is made up of two sister chromatids. Here. Two sister chromatids, which is held together by the centromere, the primary constriction. Okay. A small disc-shaped structure at the surface of centromere is called as kinetochore. Protein which is kinetochore. These structures serve as a site of attachment of spindle fibers. I told you, spindle fibers attaching to the kinetochore. Attached to the kinetochore. To the chromosome and then move to the position by the center of the cell. That is metaphasic plate. Hence, the metaphase is characterized by the all chromosomes coming to the lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fiber. Understood? So all the chromosomes are towards the center. One kinetochore will have one spindle fiber attachment here. One kinetochore will have one spindle fiber attachment like this. Here. Attachment. Okay. From one pole and sister chromatid of connected by the kinetochore of the spindle fiber of opposite pole. So one kinetochore from one spindle fiber, one more kinetochore from one more spindle fiber from the opposite pole. This is the pole 1, this is pole 2. From pole 1, one spindle fiber, from pole 2, one more spindle fiber. So two different poles, two different spindle fibers, one chromosome. One chromosome. Understand? Now, it's called as metaphasic plate. Metaphasic plate. What is happening here, students? Now, spindle fiber attached to the kinetochore, chromosome move to the spindle equator and get aligned on the metaphasic plate through spindle at both poles. Both the poles, spindle fibers are attaching. Done. Understood all of you now? Any doubt here? Okay. Now, let's go to the next stage. That is going to be transition to the metaphase. Complete alignment of the equatorial plate called as metaphase. Metaphase. Next one is anaphase. Next one is anaphase. Understand? Anaphase is the most important one. Very easy, but very easy. But understand. Anaphase. What is happening in anaphase? Understand. Okay. First, I will draw the diagram. I'll show you what exactly happens. Then I'll draw the diagram. This is the chromosome right now. This is the chromosome right now at the metaphase. Right. For example, can you see this chromosome right here? This chromosome right here, I am drawing it like this now, vertical, just to show what is exactly happening in anaphase. Okay. What is this? This is the centromere. Centromere. Now, imagine this is the chromosome. If I start pulling from this side and this side, if I start pulling from both the sides, what will happen? If imagine this is the chromosome right now. Okay, two different sister chromatids. In the middle, we have the centromere. If I start to pull this sister chromatid from this side, if I pull this sister chromatid from this side, will they split open? Yes. That is what is happening here. This particular chromosome will split into half. The centromere will split open and the sister chromatids will completely separate out. Now let's draw the diagram. One more diagram. In the corner, what do we have? We have the centrosome. Now we will draw the You see, but it was in the center first. Now it is being pulled apart. It is being pulled apart now. It is being pulled apart. That is what is happening in the anaphase. So they'll ask you lots and lots of questions. Many questions will come from here. The splitting of centromere in mitosis happens at which stage? Simple question every single year. The splitting of centromere in the mitosis happens at which stage? You're supposed to be anaphase. Anaphase. Okay. So what is happening? First of all, let me write here what is happening, students. First of all, it is the shortest phase. Shortest phase. Here we have cystochromatid of a chromosome split. Just separate, or separate. They're separating. They're separating. Okay, they're separating out here. Now here, tell me, in this particular cell, there are right now 46 chromosomes in the starting, I told you. And each chromosome has 2 chromatids. So 92 chromatids also were there. Now, but since the centromere split open, right? Can I call them daughter chromatids now? Daughter chromatids. Yes. The sister chromatids are becoming daughter chromatids. A promotion is happening. Promotion. Okay. Daughter chromatids. Now, this daughter chromatids can also be called as daughter chromosomes. Now, you might ask me why. Why can I call these daughter chromosomes chromatids as daughter chromosomes? Why? Because these daughter chromosomes will be present in the future daughter nuclei. When this entire cell divides into half, each chromatid, understand here, each chromatid will act like a chromosome. So in the case of anaphase, each chromatid will act like a chromosome, right? Okay. So cleavage has to begin in this phase. Yes, it starts, but understand the main point here. Each chromatid will act like a chromosome now. Each chromatid will act like a chromosome. So technically, instead of 46 chromosomes, and I told you, each chromatid is acting like chromosome. So can I tell, temporarily, in the anaphase, there are 92 chromosomes. 92 chromosomes temporarily. Why? Because each chromatid is acting like a chromosome. Because the centromere is split open now. And each chromatid will become a daughter chromatid in the future. Each daughter chromatid will become daughter chromosome. So temporarily, there is 92 chromosomes here. 92 chromosomes temporarily in the anaphase. Okay. And it is the best stage. To study shape of the chromosome. Best stage to study. That is VIJL shapes. Best stage to study anaphase. Now, after, can you see, anaphase is splitting up. Centromere splits. Centromere split, the chromosome separate. Chromatid move towards the opposite poles. Okay. Now the last phase. The last phase is called as the telophase. Okay, telophase. What is happening in telophase? Understand. This is the cell. This is the cell right here. Now, in this cell, the nuclear envelope is reappearing. the nuclear envelope is reappearing. All the chromosomes are clustering towards one corner. Single chromatid. All of them are single chromatid. All the chromosomes are aligning at the each cluster. Right? That is, chromosome cluster at opposite spindle poles. At the opposite spindle poles, chromosomes are clustering now. Single chromatid, all the single chromatid. So technically, before we had 92 chromosomes, right? So 46 chromosomes are this side, 46 chromosomes this side. 46 chromosomes with single chromatid. Right? Single chromatid. We learned here. 92 chromosomes. 92 chromosomes are there. 92 chromosomes are there. 46 this side. 46 are going this side. Each one with a single chromatid. Each one with a single chromatid now. See, each one has a single chromatid. Each one is single chromatid. Okay. Now, since they are clustering, all the chromosomes are clustering towards one side, they are losing the individuality. Loss of identity. Loss of identity is happening. Okay. Now, what is happening? Nuclear envelope is coming back. Nuclear envelope is back in the business. Envelope is back. Why? Because this telophase is the complete opposite of your prophase. Right? Nucleolus is coming back. Golgi complex is coming back. Endoplasmic reticulum is coming back. And everything is back to the normal now. Everything is back to its normal. That is the end of telophase. End of telophase. Okay. So what happened here? At the beginning of the final stage of chirokinesis, telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. Decondensing happening. The individual chromosomes can no longer be seen, and each set of chromatin material tends to collect at each pole. So chromatin material we have now. What do we have? Chromatin material. The chromosome cluster at the opposite poles and their identity is lost. Nuclear envelope develop around the chromosome. Everything is back. Nucleolus, Golgi complex and ER is also reformed. Reformation is happening. Reformation is happening. Can you see? For this chromosome side, for this chromosome side, it's everything is clustering one together. Everything is clustering. Now, understand cytokinesis. Understand cytokinesis now. Understand cytokinesis. What is happening in cytokinesis? First of all, tell me what is cytokinesis. Cytokinesis is nothing but division of cytoplasm. Division of cytoplasm. It is different in the case of both animals and plants. In animals it is different. In the case of plants it is very different. In animals, plants, both the cases it is very different to each other. Very different to each other. For example, in the case of animals, animal cell like one balloon. This is one animal cell. Okay. In animal cell, right on the telophase, there are two nucleus. Telophase, there are two nucleus. Yes. Two nucleus with chromatin. Chromatin is there. Now, what is happening is there is something called as accumulation of microfilaments in the corner. Accumulation of microfilaments. Now, imagine if you take one balloon and if you squeeze from that side, will there be a depression formation? Yes. If you squeeze from both the sides, it will become something like this. Yes. It will try to go on the inside. Yes. This right here is called as furrow formation. Furrow formation. Furrow formation is happening. You're trying to squeeze from the outside, from both the sides. In the case of animal cell. Now, if you keep squeezing it, one point it will touch in the middle. Yes, it will touch in the middle. When that touch in the middle happens, the two different cells will separate out. So cytoplasm is completely dividing from the outside. Cytoplasm is dividing from the outside. This type of division from the outside is called as centripetal manner. It's called as centripetal manner of cell division from the outside. Cytoplasm is dividing. Cytoplasm is dividing from the outside. Now, what about in the case of plant cell? Now, if I draw a simple plant cell, plant cell will have an outer covering called as cell wall. Outer covering called as cell wall. Now, the cell wall doesn't allow for the furrow formation. How much you press, the cell wall will not bulge. You cannot go inside. Very difficult, very difficult. So, for the very reason, what happens is, for the cell division inside a plant cell, now here we have the nucleus. One second. The two nucleuses are there, telophase, chromatin is there inside. Now what happens here is, in the case of plant cell, right in the middle, there is something called as cell plate formation. Cell plate formation. This is because of something called as phragmoplast. Which is a part of your Golgi complex. Okay. Now you see what happens here. From the inside, it moves towards outside. Can you see? From the inside, the cell plate is dividing the cell. The cytoplasm is divided from the inside. The cell plate will expand, and which will result in the two cells from the inside. This type of cell division which is happening from the inside, the type of cell division which is happening from the inside is called as centrifugal. Centrifugal division, centripetal division of cytokinesis, division of cytoplasm. I believe all of you understood now. Let's see what is given here. Mitosis accomplishes not only segregation of duplication of chromosomes, but daughter cell nuclear chirokinesis, but the cell itself divides into two daughter cells. The cell is dividing into two daughter cells by the separation of, okay, what happened? By the daughter cells, by the separation of the cytoplasm is called as cytokinesis. The cytokinesis will happen, at the end of which cell division gets completed. In animal cell, it is achieved by appearance of furrow in the plasma membrane. Can you see? There is furrow formation in the on the plasma membrane. Okay. The furrow gradually deepens and ultimately joins in the center, dividing the cells into cytoplasm into two. Dividing from the outside, it is meeting and the cell is dividing into two parts. Okay. The plant cell, however, are enclosed by a relatively inextensible cell wall. They have cell wall in them, so you can't do from outside. You cannot press from outside. Therefore, they undergo cytokinesis by a different mechanism. In plant cell, wall formation starts in the center of the cell and grows outward to meet the existing lateral walls. Can you see? It is happening from the inside and towards the outside. Centrifugal manner. Okay. Formation of new cell wall begins with the formation of simple precursor, that is cell plate formation, that represents the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria, plastids gets distributed between the two daughter cells. In some organisms like chirokinesis is not followed by cytokinesis. Imagine, the chirokinesis is happening. The two cells are dividing, but there is no complete cell division. What will happen? This nucleus will divide again. This nucleus will again divide. It will divide, it will divide, it will divide, it will divide, it will keep on dividing. In such condition, we will get multi-nucleated conditions will get. If there is no cytokinesis, if there is no division of cytoplasm, you will get multi-nucleated conditions. Multi-nucleated conditions leading to the formation of syncytium. Example is the endosperm in the case of liquid. The liquid, you drink the coconut water, no? You drink. That is basically the result of chirokinesis, not followed by cytokinesis. Okay, formation of syncytium. And with that, your entire chapter is over. But before we go to the next part, I want to teach you something called as DNA content. DNA content. How does the DNA content influence? Like, what is this 2 and 2C, 2 and 4C, 2 and 4 and 4C? All that I will teach you today. So before that, I will take one 20 minutes break and I'll come back. I'll go drink some water and eat something and come. Okay, it's getting late. So 12:52, we will class resume at 2:12. 2:12 will resume. Okay, 2:12 will resume. 2:12 will resume. Okay. Well, let's take it as 2:20. I'll just go down and come. Then I'll teach you DNA content, then significance. With that, everything will be over. 2:20 will start again. Okay? let's start, students. I told class 7:20, but let's just start early. It's fine because I just ate food very early. So now we study something called as DNA content. The meaning of DNA content is that in every step of the mitosis or every step of interphase, how much amount of DNA and number of chromosomes are there. Okay. For example, students, in G1 phase, we have something called as 2N 2C content. This is the 2N 2C. This is the DNA. Okay, this is the chromosome. Now, what about in S phase? What happens in the S phase? Okay, S phase, we have 2N 4C. Why 2N 4C? From 2C to 4C, why? Because there is the DNA replication. So this is your chromosome now. Yes, chromosomes like this. 2 sister chromatids, 2N 4C. Now what about G2? G2 remains the same. 2N 4C. In the case of your prophase, 2N 4C. In the case of metaphase, again 2N 4C. But in the case of anaphase, understand, in the case of anaphase, it is 4N 4C. You might ask me, sir, why 4N 4C? How did the number of chromosomes change? Because students, if you remember, if you go to the anaphase right here, if you go to anaphase, did I tell you there are totally 92 chromosomes? We learned that there are totally 92 chromosomes here. And if N is 23, 2N is going to be your 46, 4N is going to be 92. Yes, 4N is going to be 92. So can I tell it's going to be 4N 4C? Now what about telophase? In the case of telophase, there are two things we need to learn. That is entire cell and just the nuclei. The entire cell is going to be 4N 4C only. 4N 4C. But per nuclei it is going to be 2N 2C. How? Understand. Let's go back here. If you go to the telophase, can I tell this is 46 this side? This side is 46. So this entire cell is going to be 4N 4C. That is totally 92 chromosomes are there. 92 chromosomes there. But this side 46 is going, this side 46 is going. So can I tell this is 2N? This is also 2N. This side is 2N, this side is 2N. Because half the number of chromosomes are going up, half the number of chromosomes are coming down. So 2N on top, 2N on bottom. Yeah, 2N on bottom. 2N on top and 2N on bottom. 2N. Now the DNA content was 4C. Can I tell half of the DNA content is going this side and half of the DNA content is coming down? So 2N 2C. So this 4N 4C divided into 2N 2C and 2N 2C. So the per nuclei now, it is going to be, where did we go? Per nuclei is going to be 2N 2C. We started the entire cell division with 2N 2C. We are ending with 2N 2C. So can I call this equational division? Equational division. The number of chromosomes will remain the same towards the end. Right? So this is called as equational division or DNA content. I hope all of you understood, right? I hope all of you understood what is the meaning of DNA content and why it is called as 2N 2C. You will face some questions regarding this. If you know this concept, you can answer those questions. Okay. Now understand there's something called as significance of mitosis. What is the importance of mitosis basically? Importance of mitosis. Understand. Okay. Significance of mitosis. Mitosis or equational division is usually restricted to diploid cell. That is to diploid cell. However, in some lower plants, I told you, in insects, haploid cells also divide. Haploid cells undergo mitosis. Okay. It is very essential to understand the significance of division. What is significance? Mitosis usually results in the production of diploid daughter cells. All of you know that. With identical genetic material. That is no variations. First of all. No variations. Second one. The growth of multicellular organisms is due to mitosis. Growth. For example, from a single cell, that is zygote, to a multicellular structure, that is because of mitosis. The cell growth results in the distributing the ratio between the nucleus and the cytoplasm. So as the cell is growing, nucleus and cytoplasm is also dividing, and therefore becomes essential for the cell to divide to restore the nucleo-cytoplasmic ratio. See, imagine if only cytoplasm is dividing and nucleus is not dividing, there will be difficulty. So there is maintaining it. A very significant contribution of mitosis is cell repair. Every single time you get a cut, the repair is done by repair of the new cells is done by your mitosis. The cells in the upper layer of the epidermis, cells of the lining of the gut, plant epidermis or gut lining, blood cells are being constantly replaced. Right? See, your blood cells do not divide properly. They have to undergo mitosis. Right? Mitotic division is in the meristema tissue. That is, the plants are able to continuously divide. Why? Because of meristem. In meristem, they have mitosis. So since mitosis is happening in the meristem, that is why plants can keep on dividing and keep on increasing in size. The main reason the plant's division is mitosis. The apical and lateral meristem, right? The lateral growth of the plant, shoot apical meristem, all that is because of mitosis. Results in the continuous growth of the plant throughout their life. Throughout their life, they can grow plants. Why? Because of mitosis. Significance of mitosis, a question can come. Students, anywhere, any part you have any doubt regarding any stage, let me know in the comment section. I will clear that out personally. So students, with that being said, thank you so much for joining today's class, and